Hello, my name is Raini Scherer and I'm a neural engineer. And neural engineers connect this with that. Our brain is amazing. It allows us to think, be creative, feel, move, and all the other incredible things we can do. The brain contains about 100 billion specialized cells called neurons. We don't know precisely how many neurons there are, but there are many. The number is so large that the brain cannot comprehend this anyway. Each neuron is connected to thousands of other neurons, forming a complex network, just like the internet, but with 100 trillion connections. Neurons, also known as nerve cells, are special because they can transmit and process information using electrical and chemical signals. They are not only found in the brain, but also in the spinal cord and in peripheral nerves. They basically connect the different parts of the body. There are many different types of neurons. Let's have a look at pyramidal cells found in parts of the brain. Pyramidal cells have their name because of their shape. They look like a pyramid. Neurons consist of three parts: the cell body, or soma, which is the processing unit; the dendrites, which are the inputs; and the axon, which is the output. As you can see, neurons have many inputs, but only one output. Dendrites receive information from other neurons in form of electrically charged particles, also known as ions. Ions flow along the dendrite and up into the soma. There they accumulate, and when a specific threshold is reached, some gates or ion channels open, and this stream of ions flows along the axon. This flow of ions is called an action potential, and we say the neuron fired. As you can see, the axon here splits and divides and connects to thousands of other neurons. The interface between the neurons are called synapses. This is the myelin sheath, which acts as an electrical insulator. Remember that the stream of ions moving through an electrically conductive material is an electrical current. So the axon is basically like a cable, and the myelin sheath makes sure that action potentials arrive at their destination. If this would be a motor neuron, which are neurons which send information from the brain to our muscles, then without the myelin sheath, the action potentials would not be able to travel all the way to the muscle fiber, and the muscle is not able to contract. Multiple sclerosis, for example, is a disease where the myelin sheath degenerates, and this can lead to paralysis. Imagine you can feel, you can hear, you can see, you can think, but you cannot control any muscle in your body. 1 in 6 people in the UK have neurological conditions such as dementia, Parkinson's disease, or experience the effects of a stroke. A stroke is a medical condition in which poor blood flow to the brain causes cell death. We are now here in the Brain-Computer Interfaces and Neural Engineering Laboratory of the University of Essex, where we do our research. There are several ways how we can measure neuronal activity. Either invasively, by putting sensors into the body, or non-invasively, from outside the body. Here at Essex, we measure neural signals non-invasively using electrode and these fashionable caps. The signal we measure is called the electroencephalogram or the EEG. You might know this from some of your hospital visits. You may ask, how is it that you can measure action potential from outside the brain? The reason is volume conduction. Ion currents in neurons, they propagate through biological tissues, just like waves propagate through water if you throw a stone into it. The signal is weaker and may be distorted, but we can still pick it up. This now naturally raises the question of how accurate and specific the signals are we record. I would like to conduct with you a thought experiment. Let's assume there's a football game in a stadium next to you filled with 90,000 fans. From home, you will not be able to hear an individual fan shout at the referee. However, you will hear the crowd cheer if a team scored a goal. We cannot listen to individual neurons. We only get a bird's-eye view of the activity. What we measure is the activity of millions of neurons. The brain is very complex and difficult to study. Available neuroscientific knowledge and computational and mathematical model do not allow us to describe in enough detail the causal relationships between neural activity and the signals we measure. The problem is that different neural networks can generate the same pattern in the signal. Our task is therefore to learn more about the brain and to develop systems which can reliably and robustly detect those patterns. Our research focuses on the development of neural interfaces for communication and control, neurorehabilitation, and human augmentation. Neural interfaces for communication are designed to translate patterns or thoughts into messages for computers and artificial devices. This is very helpful for individuals with severe physical disability, as they cannot use any other means of communication. Damaged neurons typically do not regenerate. Neural networks in the brain, however, can change through growth and reorganization. Neural interfaces for cognitive enhancement are designed to monitor neural processes to assess psychological states, such as cognition and emotions, in order to inform an adaptive response from the human-machine interface. Neural interfaces for neurorehabilitation guide nervous system reorganization in a targeted and functionally relevant way. In other words, an artificial device adapts to the user's psychological state. For example, with this we can develop training programs to improve driving. We are also working on a neurotutor to reduce mathematics anxiety. This can be very useful for many. This research raises many ethical questions. It is essential from an ethical accountability perspective that changes in a neural interface user's health and performance and psychology are beneficial and personalized and ensure privacy and security.